so we have talked about the general structure of viruses, characteristics of viruses. We talked about classification of viruses. And we also mentioned that in the clinical world, viruses were often classified informally by their mode of transmission. So we talked about enteric viruses, sexually transmitted viruses, respiratory viruses, zoonotic viruses. And we gave examples of each. So make sure you can review that part. Then we also went over bacteriophages, which are the viruses that affect bacteria. And we talked about the two mechanisms that bacteriophages can use to infect bacteria. So we can have a lytic infection, which is productive, meaning new viral particles will be produced. It has a burst time, which means the time that it takes for the whole infection to proceed. It has a burst size, which refers to the number of viral particles made during the lytic infection, during the burst time. We also talked about lysogenic infection. So some bacteriophages can go through lysogeny. And in a lysogenic infection, the DNA of the virus, which was called a prophage, was introduced into the host's genome, and it will remain there indefinitely, replicating as the bacteria replicates. This led to lysogenic conversion, and it could also lead to lysogenic immunity. So that the bacteria could not be infected by the same virus while infected with a particular one. Bacteriophages that can go through lysogeny were called temperate. So lysogenic immunity prevented further infections by the same virus. Also linked to lysogenic infections was specialized transduction. So keep all of this in mind as we go over animal viruses and how animal viruses infect their host. So as we go through animal virus replication, the first steps are similar. So the attachment of the virus to the host cell by attaching to, by the viral recognition sites attaching to specific receptors in the cell. Again, usually the receptors that the virus attaches to is a glycoprotein in the cell membrane. Sometimes more than one receptor is required. In the case of the HIV virus, binds to two receptors. The receptors have a function in the cell that is completely unrelated to viral infection. The receptors is what limits the host range of the virus, so that only hosts that have that receptor can be infected with the virus. So that's what limits the HIV virus to having only human hosts, is because the receptor that it binds is only found in human cells. So dogs do not contract measles from humans because dogs don't have the receptors that the measles virus binds in human cells. Okay, so that is true for most viruses. All right, so the virus is going to attach to the host cell. And the next thing is different between animal viruses and bacteriophages. Bacteriophages are going to inject the genetic material into the host. In the case of animal viruses, the entire virus is going to go into the host. So the entire virus goes in, and there's a couple of mechanisms for this to happen. And essentially the cell internalizes the virus, and once inside, the little vacuole or vesicle that is transporting the virus is going to allow for the virus to be released into the cytoplasm of the cell. And again, we'll be more specific about all this here in just a second. If the virus is going to go all the way to the nucleus, it's going to be transported to the nuclear membrane, where it's going to inject this nucleic acid. Okay, so this is called uncoding, because now the code will be removed and the nucleic acid is what's going to go into the final destination. So again, the attachment will be the same. The entrance is different because the entire virus goes in as opposed to just the nucleic acid. Okay, so this is a naked virus. This example right here is that of a naked virus, which is entering the cell by endocytosis. The cell takes in the virus. Okay, so again, this site right here is showing endocytosis or membrane fusion, which are two mechanisms that envelope viruses can use to enter the cell. So an envelope virus, remember, is the one that has the phospholipid envelope, which is the cell membrane of the last host. So essentially, this is cell membrane surrounding the virus. So envelope viruses can join the envelope with the cell membrane of the host and push in, leaving the envelope behind in the cell membrane. And now the virus is inside the cell. Endocytosis of envelope viruses will involve the envelope virus binding to the receptors in the host, and this is going to trigger the host into taking up the virus. So now the virus is inside a vesicle, inside the cytoplasm of the cell. The vesicle eventually releases the virus into the cytoplasm, and the vesicle goes back to joining the cell membrane while the virus is now inside the cell. So a couple of things here, a couple of implications. Notice that attached to this envelope of the virus are viral spikes. So now we have these viral proteins attached, I'm just making them bigger here, attached to the envelope of the virus. In the case of membrane fusion, when the envelope of the virus fuses with the cell membrane of the host, now the spikes, the proteins of the virus, are sticking out of the cell membrane of the host. That is going to tag this cell as an infected cell. So the immune system could see that those viral spikes sticking out of the cell membrane of the host and recognize that cell as an infected cell and begin to actively destroy it. Okay, same thing can happen here in the endocytosis, because the vesicle where the envelope has joined will return to the cell membrane, so the cell membrane will eventually also have the viral spikes sticking out of the cell membrane, tagging that cell as an infected cell by the immune system, or the immune system, I guess. All right, so have an idea what membrane fusion versus endocytosis of envelope viruses entails. Now, both mechanisms end up allowing the virus to enter the cell with its protein coat still surrounding the nucleic acid, which means that in order to free up the nucleic acid, now uncoding has to take place. So uncoding is a step unique to animal viruses because the entire virus goes into the animal, into the host. So there is uncoding. The protein coat is removed, freeing up the nucleic acid. Same thing here, uncoding, freeing up of the nucleic acid. And the same thing was happening right here. Uncoding, the protein code was left behind, and only the cell, the nucleic acid was entering the nucleus. So encoding is unique to animal viruses. Once the nucleic acid has been freed, now it can be replicated, transcribed, and translated, and viral genes can begin to be produced. The first viral genes that are going to be produced are going to be those genes, those proteins are the genes for those proteins that help the virus take over the host. So those will be enzymes required for replication, enzymes that may allow the viral nucleic acid to modify the host's polymerases so that now they work on the virus also. Eventually, these syntheses will produce viral particles. The viral particles will assemble. There's also enzymes. These host enzymes are going to copy the genome of the virus. Okay, now because eukaryotic cells, animal cells, have different compartments inside, it will make it, and remember that in eukaryotes, replication of the nucleic acid of the DNA takes place inside the nucleus, transcription takes place inside the nucleus, translation takes place in the cytoplasm. So now we have different places that accomplish different functions. What that means is that if a DNA virus enters an animal cell, it will have to go all the way into the nucleus to find the DNA polymerase that is going to replicate that virus and the RNA polymerase that is going to transcribe the viral genome. Okay, so DNA viruses are going to have to travel to the nucleus in order to find the polymerases needed for replication and transcription. Remember, it is the host DNA polymerase and the host RNA polymerase that are going to replicate and transcribe the viral DNA. Okay. So it's going to use the host DNA polymerase. It may have to be modified to replicate viral genome, but it will be the host DNA polymerase. All right, now there is such thing as an RNA virus. An RNA virus does not need to go in the nucleus because it will not be replicated by a DNA polymerase. So let's talk about RNA viruses. And by the way, the COVID virus is an RNA virus. So this is basically what COVID is going to do. Just need some room, that's why I'm going back. Okay, so let's review the polymerases that the cell has. A cell is going to have a DNA polymerase. And what a DNA polymerase does is it takes a DNA molecule and copies it. Okay, so it's going to replicate DNA. Okay. All right. A cell is also going to have an RNA polymerase. An RNA polymerase takes a strand of DNA and it makes a messenger RNA copy of that strand of DNA. And then later that messenger RNA will be translated into a protein. Those are the two polymerases a cell will have. This is the problem with an RNA virus. An RNA virus is going to need a polymerase capable of taking RNA and making an RNA copy of it. So that's what the polymerase RNA viruses are going to need. Notice that neither of the cell's polymerases can do that. None of them can take RNA and copy it. They can take DNA and copy it. They can take DNA and make an RNA copy of it. Okay? Which means that RNA viruses need to carry their own polymerases. These polymerases are called RNA-dependent RNA polymerases. They are also called permeases. These are viral polymerases that the virus carries with it. That's the only way that the virus can replicate itself, which means that the polymerase the RNA virus carries was made during the previous infection, because the virus cannot metabolize anything until it's inside the cell. So the virus carries its own polymerase, RNA-dependent RNA polymerase, which is going to allow the virus to replicate its RNA. Now, what about transcription? This is the deal. The same enzyme could transcribe, in the sense that transcription requires the making of messenger RNA, and the messenger RNA will be the one that is 5' to 3' from the promoter. So if the RNA virus is double-stranded, then one of the strands will be the messenger RNA strand. If the virus is single-stranded, the replica will be the messenger RNA, and the replica of the replica will be the original RNA. In other words, I know this can get complicated. Don't sweat it too much, but I just wanted to point this out, okay? So you may hear about positive RNA viruses, and COVID is a positive RNA virus. What that means is that the one strand of RNA that the virus carries as its genome is 5' to 3', the promoter somewhere in there, and then the coding genes. Okay. What that means is that when this strand is copied, the copy is going to be 3' to 5'. Okay. And it will not be the promoter here, because the sequence will be of the promoter will be complementary. The sequence will be the complementary of the promoter sequence. Okay. But when this negative RNA virus is copied, now we have again a positive RNA copy. So the virus needs to use the permease twice to make more positive RNA copies, which are the ones where the genome is written and the ones that act as messenger RNA. Okay. Again, don't sweat it too much. If this is too much, don't worry. Just know the name of the polymerase RNA carry, and know that all of these happens in the cytoplasm. There is no need to go to the nucleus. There's nothing in the nucleus for an RNA virus. If an RNA virus, it stays in the cytoplasm. Yeah. A negative RNA virus is easy because it will just replicate itself and make a positive RNA. But then it's going to have to replicate itself to make the negative RNA to be the genome. So that's just to show that RNA viruses require this multiple replication to make multiple copies of the genome and make the messenger RNA. And also to remind you, this polymerase is a viral-only polymerase. Okay, so that is RNA viruses. There's one more type of virus we need to talk about. So RNA viruses stay in the cytoplasm. They have their own polymerase. Now, the third type of virus that we need to talk about is a special type of RNA virus. This is a virus called a retrovirus. They also have their own polymerase, and these retroviruses, which are RNA viruses, but they are special RNA viruses, do travel to the nucleus. Okay, so let's talk about how retroviruses do. And HIV is a retrovirus. Okay, this is what an RNA virus does. RNA virus carries an enzyme, a polymerase called reverse transcriptase. Which is capable of making a DNA copy of an RNA. That's what reverse transcriptase does. Remember, cells polymerases can go from DNA to DNA or from RNA to DNA. Sorry, from DNA to RNA. But there is no polymerase in the cell that can take an RNA and make a DNA copy of it. Reverse transcriptase can do that. Okay? This is called cDNA, just to distinguish it from other DNAs. And then a DNA polymerase of the host is going to, because this is a single-stranded molecule, the DNA polymerase of the host will make the complementary copy strand, and now we have double-stranded DNA. What the retrovirus is going to do next is it's going to take this double-stranded DNA and insert it into the genome of the cell. So that retroviruses pretty much stay with us for the rest of our lives. DNA viruses oftentimes do the same thing too. Insert themselves and pretty much stay with us for the rest of our lives. Okay, so retroviruses are special RNA viruses. They have their own polymerases and they travel in the nucleus. So viral DNA virus goes all the way to the nucleus. Where DNA polymerase of the host is going to copy the genome, and RNA polymerase of the host is going to transcribe. RNA viruses replicate in the cytoplasm using, I did call it permease, it's replicase. Sorry about that. Yeah, I'll mention that several times just to make sure you understand and give it the wrong name. All right. Okay, so the name of the polymerase is RNA-dependent RNA polymerase. The short name is replicase. I called it permease. I'm wrong. It was replicase. Okay. Yeah, an important point here. RNA polymerases do not have proofreading ability. So an RNA virus using an RNA-dependent RNA polymerase is using an RNA polymerase, which means the virus does not have proofreading ability, which means lots of mutations. So these will be viruses that mutate a lot. Case in point, the influenza virus mutates every year because it's an RNA virus. The RNA-dependent RNA polymerase that it uses causes lots of spontaneous mutations due to lack of proofreading ability. We get mutations every year. Another case in point will be the coronavirus, which is an RNA virus. It will mutate a lot. Okay, so high rate of mutations. Because of mutation, okay. So these mutations are called antigenic drifts. The influenza virus is a perfect example of one of these viruses. Okay, so that's the entry of the envelope virus. The RNA is out into the cytoplasm, and it will be replicated, transcribed, and translated into viral proteins, which will be assembled, and the virus will then be released from the cell. The virus stays in the cytoplasm the entire time. It has no business going into the nucleus. Let me jump to retroviruses and I'll come back to these antigenic drifts and shifts. Retroviruses or reverse transcribing viruses, they carry a polymerase called reverse transcriptase, or RTs. The virus travels to the nucleus, where the reverse transcriptase will synthesize a DNA copy of the viral RNA. So reverse transcriptase can copy an RNA into a DNA. The complementary strand, this is a single-stranded copy, so the complementary strand will be synthesized by the cell's DNA polymerase. And then the viral double-stranded DNA is integrated into the cell chromosome and remains there indefinitely. Examples of retroviruses, HIV and HTLV1. Well, there's 1 and 2, HTLV. So a retrovirus can either go through a productive infection in which viral particles are produced and released, or they may just remain latent indefinitely inside the cell. They cannot be eliminated because now they are part of the genome of the cell. And our genome has retroviruses in it that we acquire over years of evolution. We even have some reverse transcriptase in us, which will not be the same reverse transcriptase that an HIV virus would use, but we do have some reverse transcriptase from retroviruses that we're carrying. So viruses have lived with us ever since human beings appear on Earth and will continue to do so. So they're not going away. Okay, so this is the influenza virus. It's an RNA virus. And because it's an RNA virus, it uses an RNA-dependent RNA polymerase, and it is subject to mutations. This is the story of the influenza virus. There are different types of influenza virus. We have human influenza virus, which does not infect birds. We have, or other animals, except one, which I'm going to mention here in a second. We have the bird influenza virus, which infects birds, but it doesn't infect humans. Turns out both bird and human influenza virus, you probably heard of the bird flu, both of them can infect pig cells. So the pig is called the mixing vessel for both these viruses. So pig cells can house both kinds of influenza virus, the bird virus, the bird influenza, and the human influenza. So if pig cells get infected with both kinds of viruses, both viruses will begin their replication, and these produce a productive infection, so viral particles are produced. What if, by accident, one bird virus encases in itself a little piece of the human virus, or a human virus takes with it a little piece of the bird virus? Now we have what's called an antigenic shift. So this is in addition to the mutations or the drifts that typically happen. So this is called genetic reassortment. So these kinds of viruses are subject to drifts and shifts. Drifts are just regular mutations that are also occurring every time the virus replicates. On top of that, within a mixing vessel, we could have an antigenic shift. We can have one of these viruses take on a little piece of the DNA, or the RNA, sorry, of another type of influenza virus. Now, it could be that the human influenza virus now, which has experienced a shift, would be able to, actually right here, the bird influenza virus, that's what I should say, that has experienced a shift, may now be able to infect a human. Okay. So that's where sometimes we get these seasons, when we get these bird flus, for example. So essentially, this is what's happening with the influenza virus. Okay. All right, so you should know what it's a drift or a shift a shift is the genetic reassortment, a drift is just a spontaneous mutation. Remember that these are RNA viruses, and also retroviruses are RNA viruses. However, these are RNA viruses that go to the nucleus because they're going to use reverse transcriptase to turn their RNA into DNA. You should know examples of retroviruses. You should know examples of RNA virus, like the COVID-19 virus is an RNA virus. Okay. So if the viral infection is going to be productive, as it often happens with the RNA viruses, we have productive infections that are going to release viral particles, as opposed to, go back here, the retrovirus, which doesn't produce typically, I mean, it can, and it probably will at one point or another, but it typically doesn't produce a productive infection right offhand. It becomes just latent or hidden infection. Okay, so the productive part will require the assembly of the viral particles and eventually the release of the viral particles. Naked viruses are released when the host dies. Envelope viruses, and by the way, they die, the host dies because the virus initiates cell suicide called apoptosis, that's programmed cell death, so accelerates the program of death on the cell. Other viruses, envelope viruses, I should say, are going to be released slowly by what's a process called bodying of the viral particles. So, like you can see here, these little buds coming out. So essentially, the viral particles begin to push. Oh yeah, before that, these spikes of the virus are introduced in the cell membrane. The viral particles begin to push against the cell membrane and eventually push it enough to take it away, and the virus is bought out, is released out with the envelope around it. The advantage of bodying is that it doesn't kill the host cell, so the host cell can continue shedding viral particles. That should say replicases, not permeases. Permease has to do with bringing in sugars into the cells. Okay. Okay, so the problem with animal virus is that any strategy for treatment is going to disrupt the cell that houses the virus. Now, our immune system does have mechanisms to put the brakes on a viral infection. Antiviral proteins, like interferons, which technically speaking, interferons are not really antiviral proteins, but they're proteins that alarm cells into the presence of viruses and trigger them into making antiviral proteins. But antibodies, those are immune proteins that will definitely attack a virus or attach to a virus. Because the viruses live inside the cells, and they have very few viral components, antiviral drugs are difficult to come about. They're few, they generally have poor selective toxicity because you're going to have to oftentimes kill the cell to kill the virus. Viral infections can come in two types, acute or persistent, and persistent can be chronic or latent. So acute infections are easy viral infections. These are productive infections. They typically have a rapid onset, short duration. These are productive infections, and you typically are going to get long-lasting immunity from the infection. If this is a naked virus, it will cause lysis of the host cell. If it is an envelope virus, they may bud outside, releasing viruses. Symptoms are typically due to the damage of the cell as it is going through the infection. Example of acute viral infections, the hepatitis A virus causes an acute viral infection. Rhinovirus that causes the common cold is an acute infection. A persistent viral infection can be chronic or it can be latent. Either way, this is a condition that lasts for years or in a lifetime, and it may or may not have symptoms. Viruses that exhibit both acute and persistent infections, like the HIV, are also possible. So a persistent chronic infection. This is an infection in which there is continuous low-level production of viral particles. So this will be the antigen. So all through the infection, and the infection lasts typically a lifetime, the patient will be positive. Epstein-Barr. Epstein-Barr is a latent infection. Yes, it could become a latent infection. Absolutely. Yeah, it may not have to be, but it could definitely become a latent infection. That's a DNA virus. So the deal with a chronic is that the person will test positive for the antigen forever, because these are typically infections that the patient seldom gets rid of. Okay, but it will also be positive for antibodies. Okay, because the virus is present continuously, and the immune system will also be continuously making antibodies. Example of a persistent chronic infection: hepatitis B virus and hepatitis C virus. So this is persistent chronic. Epstein-Barr virus is persistent latent. Okay, so, and I'll show you the difference between chronic and latent. This is the difference between hepatitis B virus and hepatitis C virus. Hepatitis B virus, just go ahead and clean this out. 80% of the infections with hepatitis B virus are going to be acute. 20% will be chronic. But even though it's the minority of the time that the virus crosses a chronic infection, we still call it a chronic hepatitis virus. Just remember that most of the time, by far, it will cause an acute infection. On the other hand, hepatitis C is exactly the opposite. 80% of the time, you're going to end up with a chronic infection, 20% of the time, an acute infection. So both hepatitis B and A and C are considered chronic hepatitis, versus A, which was an acute hepatitis. Chronic is defined as lifetime. This is a lifetime, essentially. But again, let me just emphasize, I'm just emphasizing, most of the time, hepatitis B will give you an acute infection. An acute infection lasts for about 2 weeks, 2-3 weeks. There is such thing as a longer acute infection, which will be the ones you're talking about, maybe 6-8 weeks. Encephalitis, for example, like West Nile virus, are typically a prolonged acute infection. It will not go away in 2 weeks. It could take 3 months for us to get rid of the virus. But we'll either get rid of it or it kills us. So it's not going to become chronic. Whereas hepatitis B could become chronic. Chronic means pretty much a lifetime. Hepatitis C is definitely chronic. Without treatment, it is a lifetime infection. Even with treatment, it's a lifetime infection. All right, persistent latent. This is a different story. First of all, these are going to be DNA viruses. And what they're going to do is similar to a lysogenic infection. They're going to go into the cell, their DNA, they're going to go into the nucleus, they're going to replicate their DNA, and a replica of the DNA is going to insert itself into our genome and just going to stay there forever. The replicated DNA of the virus is called the provirus, the ones that remains in our genome as a silent host. Now, this is the difference. I'll come back to this in just a second. During a latent infection, there's typically an initial infection which goes away, and it looks like if it was acute, but it didn't. The virus would remain hidden inside the cells, and years later, it will reappear. During the time that it is hidden, if you test for the presence of the virus, if you do an antigen test, the antigen test will be negative. If you do an antibody test, that one will probably be positive. Okay? If you test for an antigen in the middle of an infection, then you're going to get a positive antigen and a positive antibodies. Okay. Whereas a chronic infection, you always get the antigen present. The antigen is not hidden. It's there. It's just acting very slowly. A perfect example of a latent infection is the chickenpox. The chickenpox virus is a herpes virus 3. The initial infection is called chickenpox. Then the virus may go hidden. I'm going to say may. Some people get rid of it. Some people don't. So the virus may remain hidden inside our cells. And 60 years later, the virus may come back as shingles. In that case, we call it varicella zoster virus. It's the same virus that gave us the chickenpox when we were children, but now it's giving us shingles as adults. Okay, so latent viruses cannot be eliminated, and the symptoms can be reacted at different times. And oftentimes the reactivation symptoms are different from the initial infection symptoms. Epstein-Barr virus is a latent virus, or it can become a latent virus. This is the deal, though. Most of the time, it doesn't. If we are exposed to the virus when we're young children and our immune system is at its best, we will never know that we were infected. Unless we do an antibody test, and in which case we'll have possibly positive antibodies. However, if we get the virus when we're teenagers, then we can end up with mononucleosis. If we may get the virus as adults, we may end up with chronic fatigue syndrome. Okay, so, and typically if we get it as adolescents or adults, it will become latent in that it may not go away. Okay, so be able to explain the difference between a latent infection and a chronic infection and an acute infection. Okay, viruses and cancers. Because viruses go into some viruses, I should say, because some viruses introduce their genome into our genome, they could disrupt genes and cause cancer. So that idea was studied, or that was a theory once we began understanding how viruses worked. The other possibility is that a virus may carry genes that cause cancer with them, or that they will turn on genes that cause cancer, from our genome. So we carry oncogenes, we carry genes that cause cancer, but they're turned off. A virus could insert itself in front of those genes and turn them on. So some viruses are known to cause tumors or cancers. The genes that cause cancer are called oncogenes. Viruses may carry their own oncogenes or may activate oncogenes already present in our genome. So oncogenes are genes that allow our cells to replicate uncontrollably, and were the genes that were working during embryonic life when our cells were replicating like crazy. And not only replicating like crazy, but these cells were able to migrate from one place to another, which is something that normal cells don't do. Just getting out of one place and going somewhere else. Cancer cells can do that. They can metastasize. Many of them can, but that's because they are regulated. They're essentially what the proteins that allow them to that are these cells have had oncogenes turned on in their genome, and now they can begin to do these behaviors that fetal cells had, but were during development eventually were turned off, because we don't want liver cells all of a sudden to grow uncontrollably or dislodge from the liver and begin traveling somewhere else in the brain and creating another liver. So oncogenes are being turned off as we developed from embryos to fetus, and they're supposed to remain turned off for the rest of our lives. But we could have a virus turning them on, or we can have a virus carrying their own oncogenes. Okay, so most cancers, however, are not caused by viruses. Only 15% of human cancers have direct correlations with viruses. Okay. You should know the viruses that cause cancer. Herpes simplex virus 4 causes a type of cancer of the blood vessels called Burkitt's lymphoma. Herpes simplex virus 4 causes another type of cancer called Hodgkin's disease. These are diseases of the cancers of the lymphatic system. This is the Epstein-Barr virus, or the mononucleosis virus. Herpes simplex virus 8 causes a cancer that's called Kaposi sarcoma, which is a cancer of blood vessels. Human papillomavirus causes cervical cancer, and we do have a vaccine available for this virus. HTLV1, are you describing TNF, the turning off and on of oncogenes? Tumor necrosis factors? No, we're not going to describe them. Yeah, no, we're not going to describe them. We may talk about it during the immune system chapter, but not in this chapter. Yeah. You're talking about TNFs, tumor necrosis factors, turning genes on and off. Yes, we're just talking about it in general, that oncogenes can be turned on or off. Yes, so they could be turned on by TNFs that we carry. It could be that the virus carries oncogenes and our TNFs turn them on. So we'll talk about tumor necrosis factors. We mentioned them in the immune system chapter, but we're not quite thrown in there yet. But yes, that's basically what we're talking about. What is going to turn that gene on and off is going to be a TNF, whether those genes are ours or they came from viruses. And viruses could turn on TNFs too. The HTLV1 and 2 are retroviruses that cause leukemia. Human T cell leukemia virus. That's what the HTLV stand for. Then the hepatitis G viruses cause liver cancer. Hepatitis V virus, hepatitis C virus. We have a vaccine for the hepatitis V virus, not hepatitis C. So we have a vaccine for hepatitis A and hepatitis B. Okay. So what I have as viral diseases are the herpes viruses and the human papilloma virus, just because they're so common, such common viruses. I will put together a short couple of slides on COVID, and we'll go over it next time, and we'll tie it to the immune system. Okay, which the COVID is an RNA virus. Okay, so the human herpes viruses are DNA viruses, and all of them are latent viruses. The HSV1 is the one that causes cold sores. And one of the points I want to make about these viruses is how common they are. HSV1, over 80% of the population in the world carries that virus. Anybody who's ever had a cold sore has the virus. So very few people has not had cold sores. So most of us carry the virus. It has in the trigeminal nerve ganglia, and that's where it lives as a latent virus. we pass these viruses on from one person to the next. That's why we all have them. They could also cause genital herpes, but they usually don't. The actual virus that causes genital herpes is the HSV-2 virus, so the herpes simplex virus. The human herpes simplex virus 2 causes genital herpes, which is an STD, extremely common STD, one of the most common STDs that we have. They spread through sexual contact. A fourth of the US population over 30 is infected. As of 2010, 16% of the entire population, not just adults over 30, but entire population is infected. It causes blistering lesions. Most of the time, however, is hidden. And acyclovir, which is a nucleoside analog, helps to keep it hidden. All right, so that's the herpes simplex virus 1, causing the little cold sores. And that's the trigeminal nerve ganglia, which is located right in front of the ear where the virus hides. HSV-3 is the varicella zoster virus, which causes chickenpox and shingles. We do have a vaccine, and we have actually a vaccine for both chickenpox and shingles. Keep track of viruses that we have vaccines for. HSV-4 is also called the Epstein-Barr virus, causes mononucleosis, causes nasopharyngeal carcinomas, a form of cancer, Burkitt's lymphoma, which is another cancer, Hodgkin's disease, which is another cancer form of the lymphatic system. So this is a virus that infects the lymphatic system, and it could become chronic, especially if herpes, we don't get rid of it because it's hidden in our own cells. It will remain there pretty much forever. So that's the reason why it's so difficult. We had a vaccine for many years that didn't really work. Apparently, they have a new vaccine nowadays that appears to work better. But the previous herpes vaccine, the shingles vaccine, did not work. It was not a very good vaccine at all. The chickenpox vaccine should prevent you from getting herpes because it prevents the chickenpox virus as a child from setting house on your body. And so if later on you get the shingles vaccine, then, especially if you have a good shingles vaccine this time, then you should be protected from shingles. But the reason why they're so difficult is because they're hiding inside our genome and they're not doing anything. So it's not active. What are you going to target? The own DNA? In the hopes that one of those will be the viral DNA? There's nothing to target. That is the problem. So that it's just a piece of DNA hidden amongst your own DNA. So until it becomes active, and then you can use acyclovir to stop the infection or to cut it short. But other than that, there's nothing to target. That is the problem. So this is a little boy with Burkitt's lymphoma. Again, notice that these are all the herpes simplex virus 4, the Epstein-Barr virus, is a lymphatic system virus. It attacks the lymphatic system. So it causes lymphoma, nasopharyngeal carcinoma, Hodgkin's disease, again, is another form of lymphoma. These are the abnormal cells that are created that we see by the virus, and we see them, they're called atypical lymphocytes. We see them with these Epstein-Barr virus infections. HSV-5, cytomegalovirus, is another one everybody has. We call it the CMV virus. And 85% of the population is CMV positive. We all carry the virus. Our immune system takes care of it, and even if it's dormant, we keep it dormant. Our immune system is able to keep it in check. So the only time that the virus comes out is when we're immunocompromised, when we're severely immunocompromised. So we did not even know the existence of the virus until we began to have severe immunocompromised patients, as in HIV patients or cancer patients or transplant patients. So once we had severe immunocompromised patients, that's when we began to find out about these viruses that we didn't know about, because we all had them, but they were hidden and our immune system took care of them. So pretty much everybody's CMV positive. So unless you are, again, premature babies, that was another population that would get these viruses. Immunocompromised patients get infections with CMV. Kaposi sarcoma, this one is one that causes the blood vessel, cancers of the blood vessel. Very, very rare form of cancer that typically affects men of Mediterranean descent who are over 60. And then, so it's a type of cancer that most physicians never see in their entire careers. In the early 80s, there was a doctor in New York City who began to see cases of Kaposi sarcoma in very young men. And he immediately thought this is something going on that was unusual. These were AIDS patients. He didn't know that at the time. We didn't know that. And because an AIDS patient is immunocompromised, severely immunocompromised, an incredibly common virus that is everywhere and we're pretty much all exposed to, but our immune system takes care of it, causes an infection on severely immunocompromised patients. So this is one of the infections that is common in AIDS patients, the Kaposi sarcoma infection, the CMV infection. Okay. Finally, the human papillomavirus. We have a vaccine for this one. It's a small double-stranded DNA virus. It causes an infection of epithelial cells, oftentimes in mucous membranes. Some strains are associated with cervical cancer and penile cancers. And we have the vaccines for both. And the vaccine protects us from the most common strains of the virus. So they cause genital warts, but those genital warts could degenerate into cancers. Okay. So that is it for the viruses for chapter 13. Again, we'll put a couple of PowerPoints on COVID and we'll introduce them into the immune system and look at it from that point of view. So we can start chapter 14, which is the non-specific immunity, next time. It's not a very long chapter. We should be able to cover the whole thing on one sitting and maybe even start with chapter 15. Okay, any questions? Okay. If I don't put up the recording for this chapter tomorrow morning, send me an email and remind me. Okay, it does take time for this thing to download, and then to upload into YouTube is going to take time also. So if I forget